Hi everybody, my name is Garces. I'm a third-year master's student at the MD Anderson UT Health Graduate School of Biomedical Sciences, and today I'm here to talk to you about combining proton radiotherapy with IDH1 R132H inhibitors to treat glioma stem cells. Now first, to start off, glia are cells in both the central and the peripheral nervous system that support axon synaptic activity. And gliomas are cancers of these glial cells, the two most common of which are astrocytomas as well as oligodendrogliomas. Now, we talk so much about gliomas because it is the most common form of malignant brain cancer in the United States today, as you can see right here. Now, when a patient comes into the clinic presenting with gliomas, the first thing that happens is that their tumor is graded on a classification from grade 1 to grade 4. Grade 1 is the most benign, while grade 4 is the most aggressive and carries with it the lowest patient survival rate. Now, within grade 4 tumors themselves, there's primary glioblastoma and secondary glioblastoma. Primary glioblastoma tumors affect primarily the elderly population and consist of the majority of glioblastoma cases. These tumors develop de novo with no known precursors. Essentially, you have a spontaneous glioblastoma tumor that forms. Secondary glioblastomas, however, are a little bit different. These constitute 10% of all glioblastoma cases, primarily are in younger adult patients. And essentially what happens is that lower grade gliomas actually malignantly transform into the glioblastoma multiform to form secondary GBM. And you can see the differences between secondary and primary GBM right here is almost a factor of 2. And so for the majority of the presentation, we're going to be focusing in on the effects of lower grade gliomas we're going to be focusing in on genetic alterations of lower grade glioma as a form of therapeutic intervention to inhibit this malignant transformation. The first of which is mutations in IDH. So isocitrate dehydrogenase, specifically the isozyme IDH1, normally converts isocitrate into alpha-ketoglutarate, as you can see right here. However, in lower grade gliomas, what ends up happening is that the IDH1 is mutated, specifically IDH1 R132H, as you can see right here. What ends up happening is that alpha-ketoglutarate is further reduced into 2-hydroxyglutarate, and the NADPH that is produced ultimately ends up being oxidized into NADP+. So this 2-hydroxyglutarate has been the subject of many studies over the past decades, ever since it was discovered in 2008. And some of the results are shown in this graphic right here from Gagne et al. In 2017. Overall, there's a consensus that having an IDH1 R132H glioma is more beneficial than not, mainly in terms of the survival type. Patients with IDH1 R132H gliomas tend to survive longer. Overall, that's just that's just a clinical consensus. And the molecular mechanism as to why this is because IDH1 R132H tumors generally have lower or higher levels of reactive oxygen species, impaired double-strand DNA damage, repair via homologous recombination, as well as DNA hypermethylation, which leads to the GSIM phenotype. And so, yeah, this leads to generally a better prognosis for IDH1 R132H patients compared to the IDH1 wild type. So the second alteration that I want to talk about is loss of the ATRX chromatin remodeling protein. Normal function of ATRX is to recruit the death-associated DAX protein, recruit histone 3.3, and deposit it at telomeres and pericentromeric heterochromatin. Now, ATRXLoss itself has been associated in the past with impaired NHEJ, impaired DNA damage repair via non-homologous end joining, as well as increased alternative lengthening of telomeres phenotypes. However, this genetic alteration is still pretty novel, and so its function is still being elucidated. ATRXLoss is specifically presented alongside IDH1 R132H in astrocytoma patients. Now, it's important. Now, we talked about these two genetic alterations, and they're very important. However, we also need to focus in on not only the tumor mass, but also the glioma stem cell population, because the cancer stem cell population contributes significantly to patient prognosis. So cancer stem cells are undifferentiated tumor-initiating cells that are extremely chemoradiation resistant, immunoradiation resistant, as well as they can differentiate into basically any cell within the tumor mass. And they ultimately end up promoting vasculogenesis, metastasis, and tumor recurrence in the patient. Cancer stem cells are known to be one of the leading causes of tumor recurrence in patients thus far. And so it's important to not only treat the tumor mass itself, but also the cancer stem cell population when developing novel therapeutics. And one of the ways we're doing this is through proton radiation therapy. So as you can see down here in this graph, proton radiation therapy compared to conventional X-ray therapy is actually more targeted for the tumor compared to X-rays. You can see right here that there's a lower entry dose for protons compared to X-rays, and that's due to the fact that protons have mass and they have what's called linear energy transfer, which ultimately culminates into what's called the Bragg peak, the maximum energy that's deposited towards the fall off of this energy that's deposited by the proton as it moves along the path length. Additionally, protons are known to induce more cluster double-stranded DNA damage than X-rays, which leads to their higher relative biological effectiveness compared to X-ray therapy alone. Additionally, the second therapeutic modality that I want to focus in on is IDH1R132H inhibitors. So there's two inhibitors specifically I'm going to focus on, AG120 and AG881, because these two are currently in phase 1 clinical trials for lower grade gliomas at UCSF. Now in vivo, these two inhibitors have been shown to decrease or inhibit 2-hydroxyglutamate production, thereby promoting differentiation of the GSC population, the glioma stem cell population, as well as impairing glioma proliferation. However, what I want to focus in on is what happens when we combine radiation with these inhibitors. So that's where my objective comes in. I'm going to be focusing on today on the results of combining these inhibitors with proton therapy, leading to what happens in terms of the self-renewal capacity of GSCs. And I believe that combining these inhibitors with proton therapy is actually non-beneficial to the patient, and ultimately probably will end up harming them in the long run, because we believe that this combination modality will promote glioma stem cell self-renewal. The way we quantified self-renewal is we used the limiting dilution analysis model developed by Hu and Smith et al. 2009. So what this assay is you see different seeding densities, in my case 20, 10, 5, and 1 cells per well, of single cell suspension of GSCs. You radiate these cells, wait for spheroids to form after 4 days, and then analyze the cells. Essentially, you quantify the number of wells that formed spheroids compared to those that did not. And as you can see right here, we believe that radiation therapy alone will impair self-renewal, while radiation combined with IDH1R132H inhibitors will promote self-renewal. And so that's exactly what we saw. We saw in the MGG18 cell line, which is cell that harbors IDH1R132H, combining X-ray therapy and ivosidenib actually does promote self-renewal capacity of IDH1R132H GSCs. Now, the graph right here that you see is a log fraction of non-responding wells compared to the dose of cells that was seeded in the first place. The more horizontal the line tends towards, the solid line tends towards, the more effective the treatment modality is. The more vertical the line leans towards, the less effective the treatment modality is. As you can see right here, the X-ray therapy line leads towards more the vertical. The combination of X-rays and ivosidenib leads more towards the vertical. Therefore, it's not as effective as X-rays alone. And we saw the same results with proton therapy. And although the result was not significant, we believe that this is still unique. This is still important, because it shows that combining these inhibitors with radiation therapy either doesn't give you any added benefit or actually hurts you in the long run by promoting self-renewal of GSCs. We did this also in the TS603 cell line and saw similar results to X-ray therapy combined with ivosidenib. So you can see right here, the line of combination therapy tends more towards the vertical, therefore it's less effective than the proton therapy alone. And also you can see right here, in terms of the neurosphere formation frequency fold reduction, is much higher for proton therapy compared to the combination therapy. Now, in terms of ATRX, I wanted to take a look also at ATRXLoss, because ATRXLoss presents with IDH1R132H in astrocytoma patients. You can see right here, proton therapy, ATRXLoss compared to wild type actually does impairs spheroid formation capacity under the influence of 4-grade proton therapy alone. However, when you look at ATRXLoss in the context of IDH1R132H in the GS818 cell line, we get similar results to the cell lines that have just the IDH1R132H alone, the MGG18s and the TS603s that I showed earlier. So therefore, ATRXLoss is not enough when under the influence of these IDH1R132H inhibitors in GS818s to promote radiosensitivity. And this was also done with vorasidenib, which is AG881, the other IDH1R132H inhibitor that I showed earlier. We saw very similar results to the ivosidenib that I just showed earlier. So what are the conclusions of this study? We show that radiation therapy and IDH1R132H inhibitor does promote self-renewal in cell lines that have both IDH1R132H as well as IDH1R132H and ATRXLoss. And we show that proton therapy is the most effective treatment modality to ultimately prevent the molecular transformation of low-grade glioma to glioblastoma multiforme. And the future directions of our studies, we want to look at DNA damage capacity, as well as apoptosis and the subsequent apoptosis and necrosis of GSCs that are treated with radiation compared to the combination modality that we talked about in this study. And thank you so much for taking the time to listen to my presentation. I want to first acknowledge my lab, my PI, especially Dr. David Grosshans, which has been a great mentor throughout my time in graduate school, the members of my lab, as well as the members of the Sawaguchi lab, which have been invaluable in helping me develop, and as well as in terms of mentoring me on this project, as well as my advisory committee, Krishna Bhatt, Fata Guan, Stephen Lin, and Skiro Sawaguchi. Thank you so much for listening to my presentation, and I'll take any questions.